Okay, so we're going to be talking about Chapter 2, the innate immunity. This is the first line of defense for the immune system. So we're going to talk about anatomic barriers and chemicals that are going to be our defenses. And then we're going to talk about the complement system and innate immunity. So, like I introduced in the last couple of classes, the pathogens are everywhere, and so our first line of defense is containment, setting up anatomic barriers. This would be your skin or the endothelial layers of your lungs or alimentary canal, and they're to prevent infection. But when they fail, then you have an infection. The first step is recognition. Um, this is done in a non-specific way. So these are broadly specific effector cells with broadly specific recognition. And so hopefully they will be able to remove the infectious agent. And if they fail, then they will recruit effector cells by recognition of pathogen associated um, proteins on the surface or mediators of uh, the immune system on the surface of the pathogen. So the recognition of PAMPs will activate the cells of the inflammation system, and hopefully then you will remove the infectious agent. If that fails, so that would be things like we talked about the engulfment by dendritic cells and macrophages and neutrophils, and then the arrival of some of the granulocytes like eosinophils and basophils and neutrophils and mast cells, um, using their the proteins and chemicals inside of their granules to attack the pathogen. After engulfment, these macrophages and dendritic cells will move to the secondary lymphoid organs, where they will do antigen presentation to the naive B cells and T cells. And if they activate a B cell or a T cell, there will be clonal expansion and differentiation into effector cells, and they will remove the infectious agent. So all of that stuff downstream of transport of antigen to the lymphoid organs is stuff we will talk about later in the semester. This uh, this talk is more focused on recognition of broadly specific effectors and PAMPs and activation of effector cells by inflammation, as well as some of the containment by anatomic barriers, some of the chemical agents, um, proteins, and um, uh, peptide uh, effectors that will be able to attack. So um, there are a lot of things out there that are able to attack us. Our first barrier is the epithelial surfaces and the skin and the mucus and waxes. But if they do get past that, then we need to overcome the infectious agent um, with innate host defenses, focus on the infection. Um, so these would be phagocytic cells and producing antimicrobial proteins and peptides. So then there are other proteins in the bloodstream that are also going to help us out. So there's a whole bunch of things that can attack us. So they can come in many forms. They can be viruses, bacterias, um, molds or funguses, um, and helminths. Those are worms. So some examples of that would be viruses like measles or influenza, varicella, which causes chickenpox, Epstein-Barr, which causes mononucleosis, as well as some bacterias like uh, Streptococcus pyogenes, which is always in your throat and can cause tonsillitis, um, and Neisseria meningitis, which can cause a meningococcal infection. Um, and those are both bacteria, 1 gram positive and the other gram negative, respectively. Um, Something like Bacillus anthracis is also a gram-positive bacteria, but it would be inhaled as a spore and cause inhalation anthrax. We could also have, so that would be the mouth and the respiratory system. We could also have gut infections. So the gastrointestinal tract or the alimentary canal can be attacked by rotavirus or hepatitis viruses, um, bacterias such as the gram-negative bacterias like Salmonella and Vibrio cholera, um, um, Salmonella typhi, um, uh, Salmonella typhi causes typhoid fever. Um, uh, Salmonella very often causes food poisoning. Cholera um, causes cholera. Um, so then you can also have, um, worm infections like trichorrhea or um, some sort of gut um, parasite infection like Giardia, for instance. Then you can also have sexually transmitted diseases like Hep B or HIV. Those are viruses or bacteria like gonorrhea and syphilis are both sexually transmitted bacteria. You can also have things that are just living in um, your urinary tract, like candida, and that can also live in your throat and cause it's a fungus and can cause infections. It's opportunistic, so usually it only gets a hold of you if you're busy fighting off something else. Um, and then we can also talk about things like ex external epithelia being uh, um, infected by funguses like athlete's foot or bacteria like Bacillus anthracis again causes cutaneous anthrax. Uh, Clostridium tetani causes tetanus. Usually, you're going to get a tetanus if um, you step on a rusty nail or something like that. You can also get tetanus from animal bites, and those are gram-positive bacteria. Um, and you can also get viruses from insect bites. Um, Obviously, something like Zika was famous for a little while, or yellow fever. Those are viruses that are transmitted by insect bites. You could also get malaria, which is a protozoan from an insect bite, or a bacterium like Lyme disease or plague. Um, and those can the vector can be insect. So so uh, when they're how do they get in? You know, like we talked about this before. They could be things that are going to infect you, but be extracellular, like bacteria that are outside of the cell, or protozoa that are outside of the cell. Um, and the way we go after them is by phagocytosis, and decorating them with antibodies, maybe neutralizing antibodies for whatever toxin they have, and we'll talk about toxins in a minute, or maybe attack them with complement. Um, you could also have them on the surface of the cell, like gonorrhea is on the surface of the cell, or cholera is on the surface of the cell. Uh, um, Helicobacter pylori or H. pylori, which causes ulcers, is on the surface of the cell. Thrush, Candida albicans, many worms can live in the extracellular spaces and attack you. And the way we're going to go after them is through antimicrobial peptides and antibodies, especially IgA, because IgA can cross into the lumen of the gut. So not all um, immunoglobulins can cross uh, into the gut lumen, but IgA is real famous for doing that. So if it's a virus, or some protozoas, or some bacterias like chlamydia or rickettsia, um, they are going to live inside of the cell, but in the cytoplasm, and we're going to go after them with cytotoxic T cells or natural killer cells. Um, they could also be vesicular, so like let's say a macrophage endocytoses something like Legionella or Cryptococcus, Leishmania. They're going to interfere with the ability of lysosomes to attach to those phagosomes, and so they can just live inside of that phagosome indefinitely. Mycobacteria are really famous for this. Um, and so what we're going to do there is we're going to again go after them with T cells and NK cells, and we can also have the T cells activate macrophages, the macrophage that swallowed them, to be a little bit more aggressive in dissolving the things that are in its phagosomes, and that will be able to attack them. Um, we could also have toxins. So there are exotoxins and endotoxins and direct cytoplasmic toxins. So they would be in the cytoplasm. So exotoxins, some of the famous ones are Streptococcus pyogenes, which causes tonsillitis and scarlet fever, uh, Staph aureus, which causes boils, toxic shock syndrome, even food poisoning, diphtheria, um, obviously causes diphtheria, Clostridium tetanus causes tetanus, and Vibrio cholera causes cholera. Um, whereas you can have an endotoxin, this is a different kind of toxin, but it goes into the cell, and so E. coli is really famous for that, causing sepsis. But we could also have typhi that causes typhoid fever, or Pseudomonas, which can cause gangrene or wound infection, and Yersinia pestis, which is in the mouth of the flea, causing plague. Um, some of the ways that tissue gets damaged is actually by indirect means. So things like causing immune complexes, or anti-host antibodies, where you have things like epitope spreading. We'll talk more about that later, and we'll also talk more about cell-mediated immunity in later chapters. Okay, so move on to the chapter, uh, uh, the second video of the chapter.